So good morning, everybody, and welcome again. So thank you very much for your kind introduction. Basically, I'm going to talk about aberrant methylation of tumor suppressor microRNA in myeloma. And I always find it difficult to talk after June because he's always given excellent talk ahead of me, so there's always a lot of pressure. So anyway, the flow of the talk comprises the following. First of all, I will talk about gene-promoted methylation in cancer, and this will be followed by the pathogenesis of myeloma, and finally by the methylation of microRNA in myeloma. So gene-promoted methylation in cancer, basically DNA methylation basically refers to the addition of a methyl group to the carbon 5 position of a cytosine ring that leads to the formation of a methyl cytosine. And this process is actually catalyzed by an enzyme called DNA methyl transferase. So in normal cells, the tumor suppressor gene, if they are associated with a promoter-associated CpG island, so they are normally unmethylated and hence allow access of transcription factors. And this will allow transcription of the tumor suppressor gene. However, in the case of cancer cells, the promoter-associated CpG islands are aberrantly methylated, and hence this will recruit a number of different proteins, including the methyl-binding domain proteins and histone deacetylase, leading to the formation of compact histone configuration. And this will preclude access of the transcription factors, and hence lead to gene silencing. So the role of gene hypermethylation in the inactivation of tumor suppressor gene will be as follows. So one of the allele of the tumor suppressor gene will be inactivated by either loss of function mutation or deletion, and the other gene will be inactivated by gene hypermethylation, and together this will fulfill the Nelson's hypothesis. And next is the pathogenesis of myeloma. So assuming that the cell you see here is actually bone marrow stromal cell. So after immortalization of the post-germinal B cell, there is a plasma cell. This will home to the bone marrow and bind to the stroma, and the adhesion of the myeloma plasma cell to the bone marrow stromal cell will lead to activation of a number of signaling pathways in the bone marrow stromal cells, and this will lead to secretion of a number of cytokines inside the bone marrow, including interleukin 6 and IGF-1. And, for example, interleukin 6 will bind to the cognate receptor on the myeloplasma cells. And this, again, will activate a number of signaling pathways inside the myeloplasma cells. And this will eventually lead to survival and also proliferative advantage of the myeloplasma cell. In addition, the adhesion of the myeloplasma cells to the bone marrow stromal cells will also give rise to a situation called CAMDR, that is a cell adhesion-mediated drug resistance. This will be very important to the reason that despite that myeloma plasma cells are poor in vitro or in the test tube, are very ready to go into apoptosis. However, inside the body, they're really resistant to apoptosis despite high-dose melphalan or auto transplants. And moreover, the myeloplasma cells will also secrete a number of cytokines or molecules, including DKK1, which is actually a soluble wing inhibitor. And this DKK1 will inhibit the development of differentiation of a osteoblast progenitor cells to an osteoblast. Moreover, the secretion of, say, for example, RANK ligand from the bone marrow stromal cells will also enhance the proliferation of osteoclasts, and together we can understand why in patient myeloma there is always a predominance of osteolytic bone lesions. Finally, there will also be secretion of a number of other cytokines, including VEGF, leading to angiogenesis. So clinically, myeloma will be characterized by several clinical stages. So after immortalization of the post-germinal center B lymphocyte, there's plasma cells. It will migrate to the bone marrow, there's a homing, and initially the patient will present with an entirely asymptomatic stage called MGUS, and followed by subsequently they will evolve into symptomatic myeloma, but at that stage it's still entirely intramedullary. Eventually they will become extramedullary disease. And at the molecular level, as June has already told us, so majority of the cases, so either around half of the myeloma cases are characterized by non-hyperdiploid stage, or actually the majority of these actually carry primary translocation of the immunoglobulin heavy chain gene, and the other half is actually characterized by hyperdiploid stage. But whether they are hyperdiploid or non-hyperdiploid, by gene expression profiling, literally all myeloma patients are characterized by an upregulation of D-type cyclins. And then when they progress, they will acquire further activating mutation of the RAS gene or FGF receptor 3 mutation, and also secondary translocations of the immunoglobulin heavy chain gene and also the MEG gene, and also P18 tumor suppressor gene deletion and P53 gene mutation, and this will lead to the development of extramedullary disease. So methylation microRNA myeloma. So microRNA are actually 20 to 25 nucleotide length non-protein coding RNA that lead to translation repression of the corresponding genes by binding to the 3' UTR of these genes by sequence-specific binding. So microRNA in cancer, they can either be oncomere, that is they are involved in oncogenesis, or they are tumor suppressor mere. So the objective of the following studies will be to study the role of aberrant promoter DNA methylation of tumor suppressor microRNA genes in myeloma by a candidate gene approach. So the questions to answer will comprise the following. One, are microRNAs methylated in myeloma? And second, does microRNA methylation lead to mere silencing? Third, does the mere methylation carry biological consequence? And if so, how? And finally, what is the implication of mere methylation in the pathogenesis, progression, and prognostication of clinical multiple myeloma? So these slides actually illustrate the following MERs that we have studied, including MER 203, 44A, 44B, and C. And together, 44A and B and C are actually transcriptional targets of P53, and also MER 124, MER 129, and MER 224. So the first question to answer is, are they microRNA methylated in multiple myeloma? So using methylation-specific PCR, basically we on the left-hand panel, you see this is a methylation-specific PCR of the normal samples and also the positive control. So what you see is that all these MERs are unmethylated in normal, but they are variably methylated in different myeloma cell lines. And hence the conclusion is that these microRNA are methylated in myeloma in a tumor-specific fashion. And then does microRNA methylation lead to mere silencing? So for example, you look at MER 44BC methylation in multiple myeloma, which has been presented by my student on the other day, on the first day, as our presentation. So I don't want to go into the details. Suffice it to say that if we study MER 44BC methylation and also MER 44B expression in a panel of myeloma cell lines, some of them are homozygously methylated, that is MM, and some of the cell lines are actually entirely unmethylated, that is UU, and some of them are hemizygously methylated. So what we observe is that in myeloma cell lines that are homozygously methylated, they have a very low expression. So mind you, this is a delta CT. So for those who are not familiar with qPCR, so a delta CT, the higher the number of delta CT, that is the lower the expression. So in the myeloma cell lines that are homozygously methylated, they have a lower expression of MER 44B. And for cell lines that are completely unmethylated, they have a higher delta CT or higher expression, lower delta CT, and hence a higher expression of the MER 44B. And so what we conclude is that MER methylation actually correlate with the MER expression. Moreover, in demethylating treatment of myeloma cells, so we see that, for example, in MER 203, so these two cell lines, in these two cell lines, KMS and WL2, they are homozygously methylated for MER 203. So upon, and this is associated with a low expression of 203, and then upon hypomethylating treatment with 5-deoxycytidine, that will lead to, first of all, the methylation of the MER 203 locus, and this is associated with re-expression of MER 203 as evidenced by the qPCR. And the same actually applies to MIA34BC, so they are homozygous methylated in these cell lines, and then upon hypomethylation treatment, the MIA is pre-expressed. And the same applies to MIA124 and also MIA224. And so we conclude that the microRNAs are actually silenced by promoter hypermethylation, and they are pre-expressed upon hypomethylation treatment. And then the next question is, does MIA methylation carry any biological consequence? And if so, how? And then I would just like to draw your attention to MIA203 methylation myeloma cells. So MIA203 so the question to ask is MIA203 a tumor suppressor? MicroRNA myeloma cells. So we look at two cell lines that carry homozygous methylation and hence very low expression or absence of expression of 203 in KMS and WL2. So basically we overexpress this MIA203 into the cell lines, and what we find by proliferation assay, that's the MTT, we find that actually it leads to inhibition of proliferation. And so MIA203 carries a tumor suppressor function in myeloma cells. And then the next question to ask, what is the target gene of MIA203 mediated tumor suppression in myeloma cells? So by two bioinformatic software, basically we arrive at the same gene that is a CRAB-B, that is CRAB gene. So CREB appears to be the target of MIA203. So as you can see, there is a lot of complementarity between MIA203, the SID region of MIA203 and the 3' UTR of the CRAB gene. So we want to prove this, prove that CRAB is actually the target gene of MIA203 by a luciferase assay. So in a luciferase assay, basically we clone the CRAB 3' UTR actually free downstream to luciferase. And so upon transcription and translation of luciferase, the translation of luciferase is actually affected by anything that binds to the 3' UTR. In this case, is the CRAB 3' UTR. So in the case when we clone a wild-type CRAB 3' UTR downstream to luciferase, so what we see is that, and concomitantly we co-express MIA203, and what we find is that MIA203 actually bind to the 3' UTR of CRAB, and then this will lead to, of course, a translational repression, and this will appear as the readout that is inhibition of luciferase activity. However, if we mutate the CRAB 3' UTR and they clone it downstream to luciferase, okay, then in that case, it does not, even in the presence of MIA203, it does not bind to this CRAB 3' UTR, and hence does not lead to inhibition of luciferase translation. And so we can see that there is actually no inhibition of the luciferase activity. And therefore, in this experiment, we proved that CRAB is actually the target of MIA203. Moreover, if CRAB is truly the target of MIA203 in myeloma, then we would expect that MIA203 restoration will lead to downregulation of the CRAB protein. And so CRAB restoration of MIA203 can be done either by overexpression of MIA203 in the corresponding cell lines that carry homozygous methylation with MIA203. So what we see is that upon overexpression of MIA203 in the cell lines, that there is a downregulation of CRAB protein by Western blot. Moreover, we can also restore MIA203 by hypomethylating treatment of the corresponding cell lines by 5-ACE-2-deoxycytidine. So what we see is that upon hypomethylating treatment, again, we see downregulation of CRAB proteins in these corresponding cell lines, and hence we conclude that CRAB is actually the target gene of MIA203. And finally, we wish to understand the implication of MIA methylation in the pathogenesis, progression, and prognostication of clinical myeloma. So basically, we answer this question is actually answered based on a cross-sectional study using myeloma samples at different stages of the disease, including myeloma at diagnosis and myeloma at relapse. So, for example, look at this slide. So, as I alluded to earlier on, so the myeloma starts from immortalization of a germinal center B cell. After immortalization, they will migrate to the bone marrow. Initially, manifests as an entirely asymptomatic stage called MCAS, and subsequently, it will evolve about 1% per year. Will evolve of the patient will evolve into symptomatic myeloma. and then upon initial treatment, they will respond, and subsequently, some of them will relapse. And so this is different stages of the myeloma. So if we look at the different the panel of microRNA that we study in this cohort of patients, so we find that some of the microRNA, for example, 203, 224, and 129 are quite frequently methylated in myeloma samples at the time of diagnosis. And some of them, for example, 34A, 34B, and C, and also 124, are actually quite infrequently methylated at the time of diagnosis. Moreover, if you look at 203 and 224, the frequency of methylation is actually quite even or comparable, either at the time of MGUS, at the time of myeloma, at the time of diagnosis, and also myeloma relapse. And so we believe that mere 203 methylation and mere 224 methylation is possibly an early event that is in the pathogenesis of myeloma. And if you look at the mere 129 methylation is methylated at the stage of MGUS. However, it's far more frequently methylated in the case of symptomatic myeloma. So we suspect that this is actually mere 129 methylation is actually responsible for progression from MGUS to myeloma. Moreover, for mere 34BC methylation, we found that it is actually very infrequently methylated at the time of diagnosis. However, is very frequently methylated at the time of relapse. Moreover, in trial patients that we have a pair sample, actually my student has presented this on Monday. So the point is that for trial patients that we got the sample at the time of diagnosis and also at the time of relapse, we found that at the time of diagnosis, all these patients except one has got methylation of this microRNA 34BC. However, at the time of relapse, 6 out of this trial, 6 of them acquire methylation by 34 B and C methylation at the time of relapse. So we believe that the mere 34BC methylation is actually acquired at the time of relapse and possibly is biologically important. And if we look at the microRNA 124, what we see is that it's infrequently methylated both at the time of diagnosis and also at the time of relapse. However, the striking thing is that it's very frequently methylated in myeloma cell lines. So we believe that this is the methylation mere 124 one is actually acquired in vitro, that is during our passaging of cell lines, but it's not biologically important. So basically, to summarize, our results shows that mere 203, 129, and 224 are frequently methylated in myeloma diagnosis and hence implicated in the biogenesis of disease. 44BC methylation is infrequent at diagnosis but frequent at relapse and hence acquired during relapse. And 131 methylation is actually acquired at the time of is in vitro phenomenon. And so lastly, we wish to look at the prognostic significance of mere methylation in uniformly treated cohort of patients. Also, we look at the methylation of the panel from microRNA 203, 129, 224, and 22, and 124.1. So again, we showed that 203, 129, 224 is frequently methylated in these patients at the time of diagnosis, and some of them are infrequently methylated. And also, for one of them, mere 224, 224, it appears to impact survival. So, for example, you look at this overall survival curve. So, this is overall survival. So, you see that patients actually who carry methylation mere 224 actually has got a better overall survival than those who have unmethylated mere 224. Moreover, in a multivariate analysis for event-free and overall survival, it seems that mere 224 still remains as an independent prognostic factor for both event-free and overall survival. So to summarize the mere methylation myeloma, so we believe that mere 203, 224 are actually frequently methylated and they are an early event in the pathogenesis of disease. And mere 129 methylation is possibly responsible for progression from MGUS to multiple myeloma. And mere 44BC methylation is responsible for, is actually acquired at the time of relapse. And 124 methylation is actually acquired as an in vitro phenomenon and is only present in myeloma cell lines, but not in myeloma samples. And mere 224 methylation appears to be a favorable prognostic factor for survival. So in conclusion, tumor suppressor mere microRNA are methylated and silenced in myeloma. And methylation enhanced silencing of tumor suppressor MIA confers a proliferative and survival benefit to myeloma plasma cells. And MIA methylation is implicated for the pathogenesis or disease relapse or progression in myeloma. And finally, MIA methylation may be a prognostic factor. Thank you very much.